Welcome, everyone. We are absolutely delighted to welcome Dr. Dawn Bazely to guide us in our Ecosystems in a Jar workshop. We are exploring our relationships with ecosystems, and we thought it would be a really good idea to try to develop a personal relationship with an ecosystem in a jar. Dr. Bazely is a staple in Canadian ecology and science communication. In fact, it is impossible to think of major campaigns across Canada that Dr. Bazely hasn't been involved in some way. She has really shaped the landscape of Canadian science communication and environmental awareness. She is the winner of the Sanford Fleming Medal in Science Communication for 2022. And we are absolutely delighted that she's here to join us so that we can build our own ecosystems in a jar. So please.
Thank you, everyone, for being here. And particularly thank you. Hi, Marcia. Hi, Adam. Hi, Nicoletta. Dave is turning out to be a major partner in crime. I do like to refer to my academic colleagues of long-standing where we've tackled issues as partners in crime because I'm always doing, ever since I was an undergrad and a graduate student, what people told me, professors told me not to do. Here, it's taken, 30 years of very hard work to reach this academic space where I have other projects where I'm working with colleagues on jewelry as wearable art made by Inuit goldsmiths, like out of the biology department. Because in the past, as you all know, STEM, people like Sho and Alex and me who are trapped in biology departments, in science faculties, we are beaten down when we try to do interdisciplinary collaboration. You can do them, you can do real ones with people from all walks of life and all kind of subject areas, but you will always have a dean or a chair in the science faculty shake their finger at you like this. This is not good for your career. This is not what you should be doing. And I'm a strategic thinker. I was an angry grad student. I'm really old now, but in the 80s when I was a grad student, I was like doing everything that grad students, we were like woke back then, okay? And we were getting in trouble and talking about sexism and sexual harassment and making complaints and being threatened. We were always being threatened. I was always being told to shut up. But I figured out that I didn't want to be here for a sprint. I wanted to be here for a marathon. So now I've been a professor for over 30 years and I have avoided being disciplined. So I just urge our younger colleagues, particularly without tenure, you need people like me to create the space, right? Shoshana and Alex, to make it possible for you all to get into these tenured, more secure positions. And I'm always disappointed when senior professors who got tenure are not going down to the wire for younger colleagues, and younger colleagues are the ones taking the risks, and that should never be the case. So I was always really careful when I was younger, and I always published super boring, but I love grass and I love microbes. So I did all my fundamental, Alex and Shoshana know, Canadian Journal of Botany, Journal of Ecology, Journal of Animal Ecology, the paid places where you publish, right, within very narrow, very focused. And then I did other things on the side. And it's really amazing that now, 30 years later, I am in a SSHRC-funded project. The chair of biology is okay with me doing all this stuff. And we are bringing the social sciences and humanities into the biology department rather than me being more adventurous and going to the other faculties, because it was okay to do that in environmental studies. But full disclosure, my undergraduate degree does include a specialist in environmental studies and politics. So I always had that. I was always there, but I'm in that classic biology department. This is garden soil and rain barrel soil.
Okay.
It has got rain barrel water. You could use, I think bottled water should be fine. And then this is another one, really different looking. Check that one out.
Oh, it's beautiful.
White, that is accumulated bacteria. And we've actually got a whole bunch of handouts that will tell you what the bacteria are that as they stratify. So the most important thing I think to think of is oxygen gradients. So down the bottom is going to be less oxygen and up the top is going to be more oxygen. And you've got a water part and you've got a sediment or soil part. And the parts at the top are going to be green because they are photosynthesizing.
Right.
And you should put your jars in the light. And what we can see here is the little jar that I made with Sho and Alex's garden soil 2 weeks ago. It's already going green. You can see that, right? Now you might ask, why does that one look so different from that one? Well, because this one had a big tablespoon of table salt put into it, which made it like the ocean. So the bacteria that can survive in those conditions are adapted to salt, whereas this is fresh water. So that's why it gets different, because the soil is full of bacteria, as is the water, and what will grow and develop is what can survive under those conditions. So all you need is some water. Scoop it out of your local lake or marsh, or your rain barrel or a water bottle, soil. Shoshana, this is unused soil from your garden.
Yay! And Nicoletta, where did you get your soil?
From my garden as well.
Wonderful.
Wonderful.
Okay. Cool. I like it.
Sorry for the garden, but the water is not rainwater, because in Greece we have to see rain from many months ago. I mean, we don't have rain.
So you're going to use bottled water.
Bottled water, yes.
Okay.
Bottled water. So we'll see how that goes, because we're not sure. I'll be curious, because that bottled water is going to have salts, depending on where it comes from. It's maybe taken from a spring or something.
It is taken from the mountains. We in Greece we have many mountains.
It's going to have its own chemistry that will affect how the bacteria.
Yes, it analyzes. I mean, there are many things to analyze here. I mean, there are many ingredients.
Yeah.
It's giving you a full chemical analysis, which will be different. So once you get your bowl, you want to do two things. Put in the bottom of your jar some torn up paper, because that's carbon.
So wait. What should we do? What should we do first?
I would just dump some torn up paper in the bottom.
Right now? Like in the bottom?
Throw your torn up paper. You could either mix it up in with the soil, but I found that when I mixed it with the soil, it all kind of floated to the top. So maybe trap it down the bottom.
So we have to, as we say,
just rip it up. Any paper. These are bacteria. They need everything. Alex, would you just like to tell us a little bit, because you will know this better than me, the new tree of life, that bacteria are the most common biodiversity on Earth?
You were saying, Dr. Bazely, because I work with what I often think of are diverse things. Small. I talk about being small things, small and little things that run the world, but they're giants. The insects, we see the big diversity like us, and then there's the what I think of as small, but in fact, those are also giants. The most diversity is fungal and bacterial. If you were to look at a tree of life, of all of the lineages of life, by far the majority of them are fungal and bacterial. And then little animal one with some fungus.
For the basic column, you're going to add an egg yolk that you'll beat up and break up and just mix that in, because that's got sulfur in it. Sulfur. And that's what makes it smell smelly. Actually, mine are not so smelly now. What happens as the bacteria break down the sulfur, or the sulfur in the egg yolk can give off sulfur gas? No, it doesn't really smell of anything. I added some more egg yolks a few weeks ago. It's already there are red and green sulfur, purple sulfur bacteria, and that's what's making the purple in here. And there's non-sulfur bacteria as well. They're actually coming out at different depths. You want to spoon to about 1/3 of the way up. So originally I filled this to about here with that mixture, and give it, if you can, give it a bit of a mixture if you've got a long enough stick, but otherwise don't worry about it, it'll just settle. I just really want to make sure that everybody puts everything they've used through antiseptic, because down the bottom, I'm going to tell you, the most common bacteria down the bottom is a friendly, not very friendly bacteria called Clostridium botulinum. And you know, Botox injections is coming from that bacteria. Amazing. And botulism, when you make jams and jellies,
yes,
if you don't process the jars enough, you could kill people. I just what I forgot to do before is just, if you can just push it down, make it firm. You want to firm it up with whatever you've got on hand. Just push it down. Now you want to grab a little bit of the soil from around the edges that doesn't have so much of the egg or whatever, that didn't get the egg worked into it, and make a layer of that up until about halfway. Yeah. Now, if you have another jar, you could do another jar where you add Epsom salts or a rusty nail down the bottom, or table salt, another kind of ingredient. Okay, so once you're done with that, we are ready to add our water. And I stirred it up, but I'm going to say don't stir it up. I mean, I was just worried. I don't know why I did that, but it just took longer to settle out. So now you want to fill, once you tamp it down, after you've added your plain soil that doesn't have access to the extra yolk from the egg and touching the paper and whatnot, or the salt, just add a layer, and that creates a gradient of saltier to less salty. You're just creating different zones. That's really what you're doing. That makes sense.
How much water shall we have to add?
Yeah, good.
Fill it up to 4 centimeters from the top. Okay. Just fill it up, because we do want to see, Nicoletta, we want to see what the bacteria are doing in there. Do not let anyone that you live with, or dog or cat, get near these things, because they are super interestingly smelly. One of the things that will happen is stuff will float to the top. I don't know whether you can see it, but but it's permissible in a few weeks' time to just scoop it out. So if you look at my other ones that are really old, there is absolutely nothing. Maybe I could do that. I've scooped everything out to make it really just quite tidy. But this one, can you see there's stuff from your garden soil, perlite, floating on the top? I will scoop that out. And we want to cover it, but we don't actually want to cover it tightly, because it will explode. Because
now she tells us
oxygen will be given off. So you want to do, here's the way that I decided to do it, which is pretty good. I discovered that saran wrap actually disintegrates in the sunlight. It doesn't last. So what I've done is I found old plastic bags and I covered them up with with a rubber band, an elastic band, and I did that. I just put it on like that. And what that does is it just traps in the gases that get given off, sulfur, which is what smells like rotten eggs. That's your sulfur bacteria. And you can then just ease the lid off when you need to, off gas, or in the Gradsky column, and it stops it from being so smelly here. This one here smells of nothing, and until this is over 3 years old, and I added egg yolk and I stirred in some egg yolk to rejuvenate it and some more paper. I fed it some more because it was quite stable, and then it smelled sulfurous. And now it doesn't smell of anything. The bacteria have degraded and broken everything down. They're so good at breaking things down. That's what they are. And that is Gradsky. You want to label it. Don't forget to label it. This one actually says something like garden, and then it says S for sulfur. That means it had an egg yolk. IC is inorganic carbon from the paper, and organic carbon as well. Inorganic and organic carbon. Yeah. You just put it in the window. Make a nice little label. But it's nice to put it on a windowsill where it's getting the sun and some warmth, because the warmer the temperature, the quicker the bacteria populations start to grow and develop.
How many weeks we have to keep it as it is, we say packed?
Nicoletta, these ones are 3.5 years old.
Forever.
Wow. So as much longer as we can, right?
That's right.
In Canada right now, actually it's quite warm today. It was -2, but now it got warmer. I don't know. So if you put these outside over the winter, they would freeze, and when they thawed, the bacteria would start growing again. So like your virtual pet or your electronic pet, they do go into dormancy. They will go to sleep, and then they'll get going again. The only thing you have to do now is watch them. Liz, how's yours?
I think it's good. I only made one here from my garden soil in Guelph.
Wonderful.
But I have more of these jars at my house in Cambridge, and so I'm going to make a bunch with the Epsom salt and the table salt, and I have many rusty nails.
Exactly, and they'll all be really different, and then we can look at them.
I'm curious, how do we think is best that we can document our columns? Like, how often should we take a photo? What's the easiest way for people to submit those photos? What do you folks think?
So this was in 2020, and it says day 4. You can see that already. Do you see that Lake Ontario had this really white bacteria, and the others looked really different? I'll stop that. So that happened very quickly, but this is the most beautiful that it was. And it was on the sunlight. It was this is my favorite picture of them. And I just lined them up and I took photos.
Wonderful.
Of different days. Can you see that one?
Yep.
Mm-hmm. Yes.
And so everyone is different. So we've got the white bacteria floating. I'll find the picture in that shows what falls out at different levels. We've got greenish, we've got yellowy bacteria. You can't see the bacteria so easily in the soil, but if you shine a light, you'll see purple and green and black, and they're all different at different levels. So it will come pretty fast. The changes. So this is showing us the gradients of sulfur, more sulfur down the bottom. The sulfur will sink down, and you get this black zone, you get these greener zones, you get these purple zones, you get a green photosynthesis. And then if you really want to know Oh, yeah, yeah. So there are methane-forming bacteria down the bottom, the methanogens, and bubbles of it will smell of methane as well. We've got purple and green sulfur bacteria, and it shows you that a black layer will be the sulfur-reducing bacteria. They're all doing different things. The white layer, that white layer, remember I showed you yes. The white layer is Begiotia and Thiobacillus. So there's that white layer. You can really see that white layer, right?
I don't know. I think it's just fun with mud. And I think we need to have fun with mud more. I think we're just so in this digital world that we're not. That's why I really liked that Gordon Pass Susan, I'm forgetting her last name, quote of that brought me onto the Tamagotchi idea, is sort of what if a pond was a computer? And so that's sort of what percolated the whole Tamagotchi idea further for me, of sort of, okay, if a pond is a computer, then this is your own personal pond for your house.
I gave a talk about how the arts aren't just about making things nice and add on, but that they are the source of our human inspiration because they are done by people who know humanity best.